Well, today's going to be a real fascinating discussion. I call this my big fat panel because it's exploring the idea of obesity, what causes obesity. And we have assembled, I think, an all-star panel today of leading lights in various different categories of knowledge that have been really doing great work in exploring why we have such a rise in obesity over the years. And each of these individuals I've interviewed in the past, and we've never had them all together on one panel together. But I thought if we're going to tackle the big elephant in the room, which is causing so much dysfunction, both on a health basis as well as a financial and economic burden in this country and around the world, then we've got to really dive into the specifics as to what is really going on with these seed oil products and high PUFA products in general and their link to obesity. What is the mechanism precisely what is driving this problem? And so just a brief introduction, and again, I'm not going to get all the accolades right, in just short time, but Petro Dobromylskyj, I met you through your blog Hyperlipid, where you've been doing great work in really getting into the weeds of explaining the mechanisms. And I had a hard time following, but I had to pause it and replay, but it was very fascinating stuff with your explanation for what's going on with linoleic acid in the cells. And Dr. Cate Shanahan, of course, you've been the leading pioneer as a woman out there around the world stage, sounding the alarm of this problem for a long time. Saw you on Bill Maher's show and many other programs. Fox was at Jesse Waters you were just on, I saw, and that's amazing stuff. So a true pioneer on this topic of seed oils, and also author of Fat Burn Fix and Deep Nutrition. Of course, we got Tucker Goodrich, who's a frequent, if you are a listener or viewer of our radio show, you see Tucker on every week. He is the proprietor of Yelling Stop, the blog, and he too has been out there from a long time now talking about seed oils and sounding the alarm. We have Brad Marshall with Fire in a Bottle, and he is someone that's been on my show exploring how this linoleic acid is basically driving so much of what you call torpor, right? A state of torpor that people are in, which is an interesting angle. And so Brad has been raising low PUFA pork and low PUFA chicken experiments with really interesting results and other types of, experimentations. He's got a background in genetics and biology. And we have Dr. Ray Peat, who's joining me from by phone today. So he is also someone who's got a long history of talking about these different issues. And each person on this panel brings a different perspective. Cate Shanahan is an MD. Petro Dobromylskyj, you are in veterinarian, right? Is that your? Yeah. And so everybody has different, Dr. Peat, you have a unique angle that I think a lot of people have been fascinated by. So I really want to welcome all of you to this discussion. Thank you.
Honor to be here.
Yeah, thank you.
Very good. So let's just start where I start off. Let's start with Peter, since you, that's the order of introduction. So Peter, go ahead and tell us just, your background a little bit and set the table for what precisely is the mechanism. If we're going to make this case to the world, we've got to figure out what precisely is the mechanism for causing obesity. And maybe there's multiple layers to this question.
Yeah, I would agree very much that I think there are multiple layers to the question. And my view has been for many years now that obesity is primarily a signaling problem, and that there are layers to the signaling. At the very most basic layer of signaling, there is the mitochondria signaling to the cell what to do with the available calories. And that signaling process goes right back to bacteria, probably archaea as well, but nobody's ever looked at them. So, but bacteria, with bacteria, growth and reproduction are inseparable from reactive oxygen species signaling. And the method for producing the ROS by bacteria, we really don't know about yet. But the mitochondrium is essentially a bacterium. And from my point of view, it has brought with it the signaling system that it had when it was a free bacterium. And it uses that to talk to the cell. So that's where I ended up with on a very theoretical basis. On a more practical basis, I started low carb 20 years ago. I realized when I started low carb that there were great big glaring holes in the low carb hypothesis of what controlled obesity, and that you could look at various populations with very, very high carbohydrate intakes and there was no obesity. So I knew right from the beginning that the insulin hypothesis of obesity was very, very attractive, very practical set of advice, but couldn't be fundamentally the only explanation. So I looked at that for many years, thought that there has to be more to it. And probably about 10, 12 years ago, I sat down and tried to work out the difference in terms of how fats and how carbohydrates were actually processed in the cell and in the mitochondria. And the concept that it might be reactive oxygen species that were essential for signaling how many calories a given cell should accept, more or less fell out of those back of the envelope doodles. And it went on from, in slow steps from there, over something like 60 blog posts now, all of which essentially stemmed from those 3 or 4. So that's where I've ended up, that the seed oils signal differently within the cell from saturated fats. And the fruit oils, things like avocado oil or olive oil, are halfway house or closer to the monounsaturated fats, which again are a normal signaling component of mammalian cells. So we've got saturated fats and mixed with monounsaturated fats as a normal milieu, and seed oils mess that up. I think that seems reasonable. That's where I come from, basically. So I'll let someone else have a go.
Dr. Cate Shanahan, can you chime in with your where you start with this question?
Yeah, absolutely. So my conclusion is that a diet of seed oils builds body fat that makes people feel sick and tired when they try to burn their body fat. And because of that, we have people overeating, because we know people are overeating by somewhere around 600 to 800 calories on average, although a lot of studies underreport that, so not everybody is aware of that. But so that drives overeating. And then the other thing that it does is it changes your, it totally retools your metabolism. We're supposed to all be fat burners. We're supposed to spend most of our time, fueling on our body fat between meals. And when you have too much PUFA in your body fat, the polyunsaturated fatty acid from these seed oils, it builds up in your body fat to a concentration that exceeds like all of human history by 6 times. It used to be around 5%, now it's 30%. And that dramatically changes the ability of your mitochondria to generate energy. It pretty much shuts them down and threatens to kill your cells, right? Because without energy, cells will die within 6 seconds. They only have about 6 seconds supply of ATP energy. And so what they do instead is they reach for another fuel. And what do they reach for? Sugar, because there's always some sugar in the bloodstream. And what does that do if you haven't just eaten? Well, it'll cause hypoglycemia, right? It'll make you feel hypoglycemic. And we have an epidemic of people experiencing hypoglycemia symptoms. I can tell you this because pretty much every single one of my patients who's overweight says, oh yeah, my doctor, previous doctor, not me, said I have hypoglycemia and I have to eat, right? When I start to feel dizzy, when I start to feel brain fog, when I faint, it'll bring on seizures in some people. This is a very serious pandemic of hypoglycemia. And it's happening because our body fat makes us feel sick and tired when we try to burn it. And it makes your mitochondria, it kills your mitochondria. So I think that I've looked as far as like, where do I fit in the context of y'all's other theories here? I feel like what every one of you says is compatible with what I'm saying. Like I feel like absolutely the signaling happens, right? And reactive oxygen species play a huge role in all this. And I mean, you other guys are going to speak. You haven't spoken yet to what your theories are, but I'm obliquely familiar with all of them, and I feel like it fits into that big picture. So I feel like what I'm bringing to it is like, synthesis, for one thing, synthesis of the theories, but for another thing, kind of, it's all proof that we're all right. Everyone that understands that seed oils are unhealthy, for whatever reason, no matter how many different reasons you have, it's correct for the most part. Everyone that I've heard, I haven't heard anybody come up with any reason that seed oils are bad that is completely untrue. Right. There's some element to of truth to, I think, just about everything. But I think the major mechanism is what I've said, is that big picture. And just to give you a little bit of background, I was like reviewing like when I really got into this. I don't know if you guys know, but I went to Cornell for cellular and molecular biochemistry in the 80s, in the late 80s. And so back then I was, even though I wasn't a doctor and I didn't know about PUFA, I understood polyunsaturated fatty acids and were prone to oxidation and the spin states of oxygen that will either allow or prevent it from interacting. And so I kind of like was primed for when I finally got into this, when I was a doctor and I got sick and nothing would make me feel better, to question the idea of saturated fat as being unhealthy. And as soon as I saw there was one research article that I saw by Eva Sodergren, and it was her PhD thesis, and it was called Lipid Peroxidation in Vivo. And when I saw that, and the molecules of linoleic acid and the reactions that happened in the body, I knew immediately, because I have all this background, that all of this would affect all of our cellular membranes, our mitochondria, our fat cells, our brain cells, and all the microstructures inside of our cells. Because you can't have normal physiology when you've got reactive oxygen species flying around, and you get polymerization of the polyunsaturated fatty acids in the membranes, which destroys an area of the membrane. And this has to do with something called the permeability transition pore, where researchers have found that burning PUFA causes this thing that they haven't really identified which protein it is. Some people say it's on the inner mitochondrial membrane. Some people say it's on the outer mitochondrial membrane. Some people say it's like one protein or another. What it is that when you burn polyunsaturated fatty acids, you blow holes in the side of your mitochondria. I mean, it's a major, major problem. It's like an explosion. And the different researchers are doing their best to kind of identify, okay, this little bit and piece of like broken, destroyed, damaged, struggling to survive mitochondria has exactly this thing on it. But the big picture is what I said. This is an inappropriate fuel. It's as if we have traveled to, well, in Star Trek, they call this thing the M-class planets, right? That are compatible with life, right? And there was a recent movie, Don't Look Up or Look Up, or one of those two, about the planet Earth blew up, and in 23,000 years, the rich people end up on another M-class planet. So M-class planet, it's compatible with life. Well, we were on it. You can apply that to diet, right? We were on an M-class diet, compatible with life. We have shifted to a diet that is not compatible. That is how profound this switch to polyunsaturates from mostly saturates is because we are constantly subjecting our bodies to an unprecedented, uncontrollable amount of oxidative stress, and it just destroys life.
Very good. Well said. So, Tucker Goodrich, I wanted to let you explain how you see this problem of seed oils and obesity.
Well, first off, I want to say it's an honor to be on this panel with everybody here. I've gotten inspiration from all of them. When we talked to Dr. Peat the previous time, I mentioned that I think every time I go back and read his stuff, it's amazing to me how much I've learned in the interim and how much more I'm able to understand what he wrote the first time around that I wasn't able to get. Same goes for everybody else here. So I thank you all for the background I've gotten from all of you. So my experience started out somewhat personal. I was a little bit overweight and very sick, and almost on after doing some reading about the problems with seed oils, on a lark, I decided one day at the salad bar in my office to stop eating them. Looking at the squeeze bottles of salad dressing at the end of the salad bar and thinking that those must be the cheapest, nastiest oils known to man to make it into that salad bar in an office cafeteria. And to my surprise, my health increased dramatically and immediately. An irritable bowel disease I had been suffering from for 16 years went away in 2 days. And over the next couple of months, I lost all of my excess weight, which I had been fighting for some time to no useful effect, culminating in putting on my pants one morning to go to work, buckling up my belt, letting go, and having them fall to the floor, which was a fairly happy thing to have happen, although the tailoring bills that followed that were pretty severe. So I got curious. Why did this happen? Why was, it totally went against what we're told to do. We're told that these oils are part of a healthy diet and that we should be consuming them, and that if we do so, we will lose weight and be healthy. And I only recovered my health when I reversed that advice. So I started, at the time I was on Wall Street as a technology expert, and I was, quite adept at researching and solving problems. So I just turned that skill set to the scientific literature. And what I've found over the years, as Peter and Cate have already mentioned, it would be nice if there was one thing going on here. It would make it a much easier story to explain to people. Unfortunately, that's not the case. As Cate said, there are, these are not something that we've evolved to eat in the levels that we are currently consuming them. I came across one study that looked at protein damage in the cell from the peroxidation of omega-6 fats. And one chemical specifically they were looking at, HNE, caused 24% of the proteins in the cell to become damaged and dysfunctional. That's an amazing impact on your body. And it's not just mitochondria, and it affects pretty much every cell process around energy generation. So, and that's after you eat them, right? We also know from a number of studies that even before these fats get into your body, they have direct effects on your appetite. They cause you to crave carbohydrates and sugar. And if you look at what we are overeating, it's primarily carbohydrates and sugar. That doesn't mean that they're causing obesity. What it means is that we are eating a chemical that induces a state known as hyperphagia, meaning overeating, and that what it stimulates us to eat is carbohydrates and sugar. Sugar does have an independent effect, particularly if you drink sugar, but it's a much smaller effect than that of seed oils. So once they get into your body, they alter your fat storage pathways, and in apparently everything from yeast to nematodes, roundworms, to humans, they cause your body to aberrantly store fat. And once that fat goes into your adipocytes, your fat cells, the stored fats in your fat cells then cause the fat cells to misfunction and become insulin resistant. So it's really, it's quite amazing how many different effects these fats have on causing dysregulation of your body system. In a normal diet, in the type of diet that we evolved to eat, we have, of course, a tendency to put on fat when times are good and to lose it when times are bad. But there are limits to that up and down, right? I mean, 400-pound people don't survive very long on the savannas of Africa. And eating a food that causes that to happen would be very anti-survival there, and not having a regulatory system would be very anti-survival there. But what we are doing is inducing in ourselves a continuous state of dysregulation, causing this obesity epidemic.
So, Brad Marshall, you have a background in molecular biology. You're also a pig farmer, so you get to see how pigs gain weight, and they're monogastric animals. So you take some very interesting perspectives on this question of how seed oils and high PUFA products cause obesity.
Yeah, and I'm glad you brought that up. Those are, like I say, the fact that I've raised pigs. So I've been kind of generally anti seed oils, ever since I read The Oiling of America on the Weston A. Price blog in, the year 2002 or something. And that, so I started raising pigs in around the year 2005. And that really was one of the things that I was trying to do, because I knew that the pigs were monogastric. I also knew, based on literature about raising pigs from the late 1800s even, that says that, you can't feed pigs anything that contains too much of the vegetable oils, or you'll get soft pork. And even back then, it was known that American pork, which was almost always corn finished, sold at a discount in European markets because American pork was known to be soft. And the Europeans finished their pork on barley, which has about half of the linoleic acid of corn. And so it's been known at least since the 1800s that that these polyunsaturated fats kind of bioaccumulate in anything, or at least any monogastric that you feed them to. And so I was sort of, I was raising pastured pork, and it was all GMO free, and I was doing everything I could to, keep my pork fat to not be soft. And so I had a lot of experiences working with other farms, and also with my own pigs on my own farm. I realized that certain pigs, genetically, it didn't matter what you fed them, they always had soft fat. And so, I saw very firsthand that the saturation of your own body fat, there was a huge difference based on what you fed the animal, but also the genetics of the animal. And so there was something happening internally that could affect the body fat saturation, and then obviously externally what you fed them made a difference. And so if you read Fire in a Bottle, I talk a lot about an enzyme called SCD1, and what that does is it turns your stored body fat from saturated fat into monounsaturated fat, and then also, of course, the amount of dietary polyunsaturated fat. And so, because I think that, sure, the fat in your last meal, of course, plays a huge role in what your mitochondria are actually oxidizing, but it's sort of blended with your stored body fat before it enters the mitochondria. And so you have these two different sort of pools of fat that at any point in time you're oxidizing. And so I've spent a lot of time thinking about that, and that really is where the conversations about the torpor comes in, and and the torpor thing it's kind of an analogy. Humans obviously don't. A torpor, for those who don't know, is it's kind of an alternative metabolic state that hibernating animals use before they hibernate. But one of the things that they do is they have to eat a lot of polyunsaturated fat to be able to go into torpor. And then as they approach torpor, they increase the amount of SCD1 that they make. And so hibernating animals eat a lot of PUFA and they have a lot of SCD1. And so hibernating animals have very, very unsaturated body fat compared to an animal that is never going to hibernate, even if they're monogastrics. And so then when you look at obese humans over the last 70 years or however long, obviously polyunsaturated fats have increased, but the amount of SCD1 has also increased. And so when you look at especially in the muscle tissue of obese humans, there is an incredibly direct correlation between the amount of SCD1 produced in the muscle tissue and the BMI of that human. And so whenever you see a correlation that's that direct in the real world, I think that's interesting. It, of course, doesn't necessarily prove causation, but I've seen all of these parallels between changes in body fat saturation and obesity, suggest in the exact same way that animals who want to store fat for hibernation do it is the exact same way that obese humans behave. And so that was right. And of course, all that started with me just trying to make the pork fat firm on my pigs that I was raising, and it has sort of gone from there. And so now on the blog, I'm digging. Peter talks about he's at a very low level, and I'm sort of like one level above that. I'm a molecular biologist. I like genes and genetic pathways, etc. And one of the ways that my research keeps bringing me back to is how we regenerate NAD plus, because if you look at obese humans, it seems like we have too much. So NAD plus is it's an energy carrier, an electron carrier. It basically, as you break down your food, whether or not it's carbohydrates or fats, NAD plus picks up a hydrogen, becomes NADH and a couple of electrons, and it takes it up and it drops it into the mitochondria electron transport chain. And that's basically how an essential part of how we turn our food into ATP. And so if there's not enough NAD plus around, we simply can't run our metabolism as quickly. It becomes a limiting factor. And if people have heard of this, the Sirtuins, Sirtuins require NAD plus, and they remove acetyl groups from things. And if you look at obese humans, they have a lot of acetylated mitochondrial enzymes, such as complex 1, complex 5, a whole bunch of them. And so your mitochondria don't work as efficiently if you have a lot of acetylated enzymes, which is to say, if you don't have enough NAD plus. And you see this kind of irony in obese people that obviously they have a lot of energy. The person has plenty of stored energy, but yet their cells are still low in ATP because they're not efficiently converting all of that to ATP. And so and there's an enzyme called NNT, and so this will connect sort of what Cate and I were saying about functional mitochondria back to what Peter was saying about ROS. What this enzyme NNT does is it basically takes the ROS and it returns NAD plus. It's kind of a magical device that in the process of removing ROS from the mitochondria, we actually get NAD plus back. And so what that looks like to me is, okay, so if you could get your fat really unsaturated, then you won't make as much ROS, and that will give you less NAD plus, and that will give you the ability to store body fat and get ready for hibernation. That's kind of the short version of it.
Very good. And Dr. Ray Peat, you get to pick who was right. I'm just joking. Now give us your take on this question, please.
I've considered all of those over the years. 50 years ago, my dissertation in the biology department at the University of Oregon was looking at the oxidative metabolic changes that happen during reproductive aging. And immediately I started running into the close interaction between aging and polyunsaturated fat metabolism, producing age pigment, for example, and estrogen. Each one intensifies the other, and you can overlap those with oxygen deprivation in itself, or X-irradiation, or any sort of stress that uses or wastes oxygen. It accelerates the interaction, decomposing PUFA with aging under the influence of estrogen. And at that time, I had to see the importance of the accumulating PUFA with aging, but already the literature was starting to claim that not only is PUFA in the food good for preventing cancer and heart disease, they said, but even preventing obesity. I've seen dozens of articles, more and more over the years, throwing in prevention of obesity with the other gross mistakes. It causes cancer and causes heart disease and reproductive aging and all of the pro-inflammatory processes. And all of that comes from the immense profitability of selling products. Every time there's a super profitable product, the science follows the profit, absolutely going against the facts in the case of PUFA and estrogen, but at least a dozen other substances. And so you have to learn to disregard about 90 to 99% of the published medical and even biochemical articles as being nothing but reflections of advertising. And I started looking at the physical properties that would account for the fact that mammals in general, studies have been done on calves and human babies, at birth, they're generally so-called deficient in the essential fatty acids. Their tissues are making mead acids, taken as a deficiency of the plant-derived PUFA. And so I saw that the fact that animals generally close to 100 degrees Fahrenheit, 37 degrees Celsius, generally have that process of accumulating during their lifetime more and more of the plant-derived PUFA, and in the process, their own metabolism is being damaged and slowing down. So from extremely high metabolic rate at birth in the first 2 or 3 years, as the PUFA accumulate in the tissues, the metabolic rate slows down. Takes another big change towards inhibition of oxygen productive use at puberty, and then when growth ceases in the 20s, when you're no longer diluting those stored PUFA from the environment, the rate of accumulation increases, so that by middle and old age, the metabolic rate is much slower. The brain and arteries and other tissues are starting to fill up with the largely cholesterol esters of the polyunsaturated fats, also the stored triglycerides. All of these become more polyunsaturated with aging. And I think the basic reason for that is that the double bonds, the more the double bonds the fat has, the more affinity it has for water, and so it has a greater tendency to circulate free in the water. And in the process, those same multiply unsaturated molecules block thyroid function and release estrogen from the steroid-binding globulin, making estrogen more available, thyroid less available. But the tendency is not only to not oxidize these because they don't enter the cells for oxidation. It takes a more saturated molecule to get into the mitochondria and cells. So the free-floating free amino acids escape oxidative use and are preferentially stored as triglycerides and phospholipids and esters of cholesterol. So all of the tissues lose function with aging as they accumulate these fats, and that all along the route, the metabolic rate is slowing down. The temperature falls. It's somewhat defensive. The cold temperature plants, fish living at a cold temperature, are very highly polyunsaturated. That's necessary just for physical flexibility. A cold fish is stiff because if it has saturated fat, but the polyunsaturated fats keep it liquid at refrigerator temperature. And so the more cold organisms we eat, the worse the problem is. That was what led me to experiment with coconut oil, and its relatively high saturation goes with a higher metabolic rate. And Pamplona and other researchers have shown that the more unsaturated an organism is, by, for example, birds are much more saturated than mammals, and for a given weight, they have a much longer lifespan. Pamplona and others showed that that's a general principle, that life is shortened by the degree of polyunsaturated fats in its tissues. The physical property of being more water soluble, leading to the accumulation in the tissues, means that when you're under stress and release stored fats, both from triglycerides and phospholipids and esters, these stress-induced free fatty acids are going to be more polyunsaturated. The older you are, the higher the accumulation, the more harmful these free fatty acids become with aging. And the diabetes, obesity, and cancer, inflammatory degenerative diseases, all of these are linked through the both the presence of free fatty acids in circulation and the result of that. It's an example of the Randall cycle in which oxidizing fat stops the oxidation of glucose. And it not only shifts the metabolic ratio, but it actually lowers the metabolic rate. Old people, old men, for example, at rest, are burning actually more fat than the young man in the same conditions. But the old man is laying down fat at the same time, burning fat faster than the young man, but getting a higher percent of body fat. And you see the same thing with the waist to hip ratio, deposition of unnecessary fat corresponds to a lower oxidation of glucose and a higher oxidation of fat, or the respiratory quotient. And that's a general thing, that the respiratory quotient in diabetes and all of these stress diseases goes toward fat oxidation, high estrogen, and old age, and any kind of sickness correspond to that process. The way the mechanism behind the Randall cycle, when you're under stress, one way of keeping some energy process production going is to get rid of the excess NADH so that you can produce more oxidation, so forming lactic acid, which leaves the cell. That consumes some NADH and allows some energy to continue to be produced. But fat synthesis is the other electron sink that gets rid of excess electrons. So you burn fat, oxidize fatty acids, and you're increasing the NADH, raising the reductive properties of the cell, and to get rid of those excess NADH reductive atoms, you have to either produce more fat or more lactic acid, or both, and generally it's both of them. Diabetics have a chronically high lactic acid production as well as high free fatty acids. So it starts with the physical properties, the water solubility, and ends up with the problem of electron excess, and the making lactic acid and fat is the solution to that excess of electrons.
Well, very good. I really appreciate all of your answers, and I have another question for everybody, but before I do, I want to give everybody an opportunity. Just raise your hand or speak up if you want to ask a question to somebody else on the panel, or ask for clarification or a comment or anything. Just if there's anything that's been said so far that people want to respond to.
Well, the only thing I would like to add is on the multifactoriality of this, based piggybacking off of what Tucker brought up, is that there's so much going on here that it's actually detriment to basically the cause of helping other folks understand what the issues are.
Right.
There's like a saying in law that's like, if you want to get away with murder, make sure that before you kill the person, you rob them, and if possible, you rape them, and do as many other different things, because then the attorney can get up there in front of the jury and say, well, what is it you're saying he did? And it just gets confusing, and people just give up. And that's kind of, the problem with these seed oils.
That, which is a fair point to make, but if we're going to convince people that what we're saying here is correct, and I think it is correct, at some point we're going to have to come up with a credible mechanism to explain what's going on. And as I said, it's a shame it's not a simple one, but a lot of people in the United States are required to eat these foods essentially by law. They're in the dietary guidelines. They are fed to people in schools, in the military, in hospitals. And we're going to have a solid evidentiary case to convince the authorities that they've been wrong for the last 60 or 70 years and that they need to change this pattern. And unless we can point to the studies that show that this is what is actually happening and however complicated it may be, and it explains why, not just everybody in America or most people in America, but most people around the world have suddenly decided to overeat and get fat, right?
You can make it complicated, but I think that you can make it simple. And I think you can tell me how I'm wrong, but I think it seems pretty simple to say that a seed oil diet makes us build body fat that makes us feel sick and tired when we try and burn it because of the inflammatory effects of the PUFA in mitochondria and other tissues. But I think that's
which
about
is
as
an
simple
excellent
as you can make it.
Yeah, which is true, but that's not going to convince a scientist. He's going to want to know, okay, how does that happen?
I mean, wait a second though. I mean, why wouldn't it convince a scientist if it's true?
How do you know it's true without seeing the mechanisms?
There are mechanisms. I'm not making any of this up. There's multiple mechanisms. There's multiple steps in there. But your body fat, if you eat too much PUFA, it ends up in your body fat. That's step one. Then between meals, when your insulin is low, and it's, even if you're insulin resistant, your body fat does release free fatty acids into the circulation. Those will get picked up by the cells and utilized by the mitochondria, and in excessive concentration, that shuts down or reduces energy production so that the cells then, this is all step by step. I'm just taking you through the process of every single one of these steps is not debated. And then when this one might be a little controversial, when the cells are forced to burn PUFA, that they, instead of shutting down, they just put more GLUT4 transporters and slurp more sugar out of the bloodstream. That part is a missing step. I haven't actually seen too much evidence of that. I've seen indirect evidence of that I can point to, that the brain actually starts to tell the liver to produce more sugar to increase the blood sugar level. That's how you become diabetic. That's like another that's why you raise your blood sugar level when you're type 2 diabetic, because the brain says a normal blood sugar level of 100 isn't enough. I want it to be 110 fasting, or 120 or 30. But everything that I've said, except for that one step of, what is the exact concentration of PUFA in the body fat that makes the cells increase their GLUT4 transporters and slurp in more sugar from the bloodstream. That's the only piece that's maybe missing. But everything else is actually solid, and no one in their right mind, I think, would refute any of that. So there is science. It's just that it's more than just a soundbite, and that's the problem. We have to force our scientists to actually listen to something more than, we shouldn't have to, but we do have to, get the people who are should be in this conversation to listen to the whole argument, not just a short sentence like like cholesterol clogs your arteries or something simple, oversimplified like that and ridiculous.
Right.
Can I have a
Agreed.
Peter?
quick word with Tucker, or quick comment. I find your approach, your concept that PUFA make endocannabinoids make you eat is very soundbitey. Yeah.
Well, it's a great soundbite.
cholesterol
Cate
clogs
put it
arteries.
in her book. They give
Yeah.
you the munchies.
Yeah.
That's probably the best soundbite
Yeah.
we have.
The problem I have is that I think we are talking about a process, and I don't think it happens immediately. And there is a ton of research where if you feed PUFAs to laboratory rodents, you will invariably make them more insulin sensitive. So you do it diet-wise. But if you keep doing it, 6 months down the road, they'll be intensely insulin resistant. Their fat cells may be still insulin sensitive, or they may be insulin resistant. That depends on your study. But the initial response is that putting polyunsaturated fats into a rodent model makes them insulin sensitive. It happens to make them fat as well, but that's the function of insulin. So making them insulin sensitive, I have no problem with that making them fat. But the initial response, and in cell culture, you can feed polyunsaturates to cell cultures without doing them any damage at all. You feed pure saturates to cell culture, and there's a reactive oxygen species driven catastrophe. Particularly if you're culturing 25 millimoles of glucose, which is the standard culture medium, and you add 400 millimoles of pure palmitic acid to that, your cells will just die. But you can add 400 millimoles of PUFA to it, and there's no problem. Acutely, the cells will be perfectly okay. And any scientist that we ever try and convince will produce those papers and say, well, look at this.
And
Well,
as
that's
you
just silly, though,
pointed out, Peter,
there was a
that's not
concept.
That's just a moment. As you've pointed out, Peter, that's not a physiological model.
Right.
No, it's
Palmitic acid and high glucose.
Trotted out though. Yeah.
Right. Of course.
Yeah.
Yeah, I mean, you can point to a lot of things that are irrelevant to the argument and try to divert the argument, but we can't let them do that. We have to say, wait a second, that's irrelevant to our argument, and prove me wrong, right? Like you can say that too. Every one of you can say, you guys prove me wrong, because I want to run the conversation here. You guys have been running the conversation for a long time. We've been getting fatter and sicker. So try to, take what I'm asserting and tell me where it's wrong, because I'm totally open to that. And actually, what you said, Peter, what you brought up is that like the insulin sensitivity. That's totally in keeping with what I said. And it's indirect evidence of what I said, that when you force feed cells PUFA, they want more sugar. They're going to be drawing more sugar out of the bloodstream. Well, you might measure that. It might look like insulin sensitivity, right? So that is indirect evidence of exactly what I'm saying.
Yes. No, my concept is that we get fat because we're pathologically insulin sensitive. We're too sensitive. That's the problem that I see.
Yes,
and that is PUFA. But
I agree. It's as if obesity, we're trying to protect ourselves by building fat, right? We're trying to protect ourselves from burning PUFA by becoming obese. Right. And I've brought this up with on David's radio show, because I actually have seen a lot of, like, in my practice, it seems as though it's the people who are not, really obese who end up with cancer. And you could make an argument there. You could explain that, and not just cancer, but also autoimmune disorders. You could explain that as because perhaps they are burning the PUFA when they shouldn't be, and somebody, and therefore exposing their DNA to oxidative damage, and getting cancer. Of course, if you believe it's a genetic disease, but regardless of the mechanism, you're creating the oxidative stress by burning the PUFA instead of eating more, which would allow you to get more glucose, which is a safer thing to burn. Right, because it doesn't have the double bonds, it doesn't have the polyunsaturated fatty acids, and it's safe to burn. It's not the ideal fuel, but it's certainly better than. So so that obesity, basically overeating, is a person who's developing a habit of overeating and to protect their cells from having to burn the damaging PUFA. And so it's like a metabolic choice there that their body is making for them. And and I think that's PUFAs are so bad that people will eat themselves almost to death in attempt to not have to burn that and damage their mitochondria.
Okay.
Dr. Peat, how do you sort out these different arguments? I mean, I'm a layperson, so I'm listening, and it all kind of seems to be going in the same direction. Do you have any thoughts on that?
I think if you start trying to understand what the so-called Randall cycle is all about, you see that the interference of fat oxidation with the ability to oxidize and get energy from glucose. That's the essential problem. The faster you oxidize fats, and that's promoted by the unsaturation degree, the faster you oxidize fats, the slower you oxidize glucose. And that shifts the whole electronic balance, for example, away from carbon dioxide production and full energy use of the oxygen towards fat synthesis and lactic acid synthesis.
I think that's an overstatement of what the Randall cycle actually does. I think the Randall cycle is simpler. You can just it's really just a description of the cell has an option to burn fat or sugar, and
but the fat changes. The fat changes the ability of the cell to use sugar and to oxidize.
Well, you could put it the other way as well. I mean, they influence each other, right? You could also say the glucose has the ability to influence the fat. So it's a two-way street.
That means when you exaggerate the supply of glucose, you can actually lower the free fatty acids and protect against all of the consequences of burning too much fat.
Right. So the Randall cycle is a regulatory process that, if I understand this correctly, that determines what fuel the cell is going to use. And of course, cells can use two fuels at once, obviously, but that's kind of if you want, the seesaw that allows the cell to determine what fuel it's going to use.
But that use of type of fuel determines everything. The electronic balance regulates the synthesis of fat, and so the more fat you burn, the more fat you synthesize. The old person who is burning much more fat than the young person is still making fat for deposition. They become fatter because they are burning too much fat.
Dr. Peat?
That's well established, but it just doesn't seem right to people, and so they ignore it.
When you say that they're building, as you burn fat, you build fat, is the presumption that you're building the fat is coming from glucose because you're not burning the glucose, so it's being converted to fat? Was that your point?
When you burn fat, you increase the ratio of NADH to NAD.
And that generates the fat from whatever it can get.
So the more fatty acids you burn, the more fatty acids you synthesize.
Yeah, and so Dr. Peat, if I understand you, and correct me if I'm wrong, the argument about what is controlling the Randall cycle is really kind of an argument about redox balance. Would you say that's correct?
Yeah, absolutely.
Yeah. So can I add slightly to that? And that is, I think we can all agree that the Randall cycle partitions what you do with calories. What I was looking at from the ROS point of view was looking at normalizing the total calorie intake to the cell, and if the ROS make that input to the cell normal, the Randall cycle should simply sort out what gets oxidized and what gets stored in an appropriate manner. So total input to the cell was where my thinking on ROS was coming from. Randall cycle afterwards to sort out what's done with the input to the cell. Does that make sense?
Yes, Peter, I agree with that. I think we want to look at it like from the proper direction. So like the big picture is that when you are burning, when your body has a lot of, the big picture is oxidative stress. And having the physiology subjected to too much oxidative stress is going to have an influence on things like the Randall cycle and the NAD to ratios and everything else. But it's the direction that we're going is, it's not the Randall cycle running everything. It's the fact that we're eating so much more PUFA, and it's subjecting our cells to extreme amounts of oxidative stress, depleting our antioxidant systems. That is going to have an effect on the Randall cycle and on every other physiologic process. Insulin sensitivity, the translocation of glucose into a cell or not, whether or not the liver is able to do any of the normal detoxification. It's going to have an impact on every process. And so my point I'm trying to make is that we can't take one little process that the body normally does under normal circumstances and blame, that for everything. The big picture, we can't forget. I mean, it's kind of a little bit like missing the forest for the trees, it seems to me, because the big picture is the forest of PUFA that we're all living in. And it's just adulterating our physiology on every level. Certainly what you're talking about, but many other things. That's the direction that I see things going, and the explanation that makes more sense to me.
For almost 100 years, the oxidative stress and oxidative damage has been emphasized, rate of living theory and so on. But there's almost been 100% neglect of the reductive stress process, and that's what I'm talking about. That reductive stress precedes the reactive oxygen species production. The problem is reductive stress, and that's promoted by not enough glucose oxidation, too much PUFA.
Not enough glucose oxidation? That's not making any sense to me, because as humans, we're tooled to burn fat way more than glucose. So I can't accept that.
Ironically, I'm literally the current blog post I'm working on is literally called "Reductive Stress Causes Oxidative Stress." And so it's interesting.
Well, can you take a moment then and just define it so that the people watching this can
understand?
and
so oxidative stress. So NAD plus and NADH are a really good example of this. And so they're almost the same molecule, except one of them, NADH, obviously has an extra hydrogen. And so NADH is the reduced version of that molecule, and NAD plus is the oxidized version. And so in a system where you have, where a lot of things are oxidized, too many things are oxidized, that's called oxidative stress. And so in the NAD system, if you had oxidative stress, you'd have too much NAD plus and not enough NADH. But what you see in most obese and diabetic humans is the opposite. You have too much NADH and not enough NAD plus. And that lack of NAD plus is called reductive stress, and it is a real problem. And one of the ways that you can get oxidative stress is, kind of like I mentioned before, if there's not enough NAD plus, then your mitochondrial enzymes become acetylated and they stop working. And I think that that ends up driving this huge amount of reactive oxygen species coming out of your mitochondria, because like complex 5 is acetylated and it stops working efficiently. And so you have no way to get the, you know, the electrons entering the electron transport chain to come all the way back down to make ATP, because complex 5 isn't working. And that's when you get this kind of massive oxidative stress, because all of these electrons are trying to run through the electron transport chain and they can't do it. And so they're coming back out as reactive oxygen species. Whereas if you're not in reductive stress, if you have enough NAD+, then complex 5 doesn't become acetylated and the electrons can run through, and you don't end up with that kind of massive amount of reactive oxygen species production. But that sounds like similar to what Dr. Peat is saying about the Randall cycle, is you need, you know, if you wind up with too much NADH, then, you know, your metabolism just doesn't run right.
Yeah, and experiments show that if you uncouple the production of ATP by the electron transport chain, if you waste oxygen, run glucose through the system at a very high rate, excess site rate, for example, will uncouple and become wasteful use of oxygen and fuel. That uncoupling is consuming oxygen at a high rate, but it's stopping random reactive oxygen species production. It gets rid of the dangerous electrons. It gets rid of the harm of reductive stress. Faster oxidation reduces ROS.
Right, and the other thing I mentioned before is the other way to get rid of the reductive stress is if you're burning saturated fat. We have this system that removes ROS and it gives you back NAD+, and that's the enzyme. Well, really it takes two enzymes. It takes glutathione reductase, and then it takes this thing called NNT. And so that actually uses ROS when you're burning saturated fat to give you back the NAD+. But once you switch to burning PUFA, you're not making the ROS in the same way, and the PUFA keeps you in that reductive stress.
I'm not buying the reductive stress. Sorry, the oxidative stress. I think what's happening is that when you've got so much oxidative stress, you can have some like backup in some like aspects of the whole cell machinery, but it's I can't see how it's driving. Where's this reductive stress coming from? And PUFA causes oxidative stress. So how do we go from oxidative stress to reductive stress? The only way you can do it is by invoking like one or two single metabolic pathways as like these dominant pathways and controlling the cell energy partitioning. And it doesn't make sense to me. I'm sorry. It seems like a small picture kind of explanation. I'm sure it's something that can happen, but I just don't think it's the dominant process. And it matters because if we're trying to like say this is what's going on, we can't like finger a metabolic process that someone else, the opposition, could say, well, easily say, there's no way that process doesn't do that. Like, for example, there's an alternative explanation for why the SCD enzyme is affected by PUFA, and that has to do with trying to maintain a certain amount, trying to maintain the proper melting point of the fat. And so if you have a high PUFA diet, then people end up with more saturated fat in their body tissues than normal. And that's actually been one of the pieces of evidence that the American Heart Association uses to prove that saturated fat correlates with obesity because they find people who are less healthy, more metabolically deranged, who have been eating the higher proportions of PUFA, they have more saturated fat in their adipose tissue. And they're saying therefore it's because they eat more saturated fat, right? They're saying that that's what made them fat. And that is a completely inside out convoluted way of looking at it. And we can't do that if we're going to be right.
But
right.
The evidence is quite clear that one of the major changes in the 20th century, along with the increased consumption of seed oils, has been an increase in the adipose content of polyunsaturated fats. Eating polyunsaturates does not increase the amount of saturated fat that the body stores. If it did, then
does.
Brad,
wait,
if it
relatively,
if not relatively so at all, if it did have that effect, then Brad wouldn't have the problem with his pigs of having soft fat. Right.
Well,
the soft fat.
I'm talking
just
about
a moment. The soft fat is due to the storage of polyunsaturated fats.
So look, you take a look at the composition of human adipose tissue through the past 100 years, and when you look at the ratio of saturated to monounsaturated fat, you see that we actually have more saturated fat now than we did in the past compared to monounsaturated fat. And yes, you are, Tucker and Brad, you are both correct that now we have more melty fat, we have more PUFA in our fat, but as a way to protect against that, the body has no choice how much PUFA is going to end up in the adipose tissue, right? That's just a reflection of the diet. We can't change a polyunsaturated fat into a monounsaturated fat. We have we have no capacity to do that. But we can change
do
sugar.
actually. That does in fact happen.
All of the evidence I've seen is that there's
but
definitely
not to a big degree if it does, because that's why we have this huge increase in body fat, polyunsaturated fat. So I need
because we've overwhelmed our regulatory mechanism.
Yeah, I would say there's a lot of evidence out there that
which is very small.
in obesity and diabetes, the ratio of monounsaturated fat to saturated fat increases. If you look up
monounsaturated, total unsaturated,
if you look up the desaturase index, there's a ton of evidence that, at least specifically in obesity, I guess in diabetes too, the ratio of like oleic acid to stearic acid goes up with obesity.
Well, stearic acid is just one of the many saturated fatty acids, right? So we want to look at the total saturated fatty acid. And we have other fatty acids besides stearic that we can make. So that's what I'm talking about.
Right. And yeah, it looks to me like average Americans now have something like 10% less. Well, like in the 1960s, the studies that we have, there was one in Indiana where saturated fat in lean adults was something like 36% total saturates. And if you look at obese people now, it's something like, 25 to 28% total saturates. So it looks to me like saturated fat has decreased specifically in obese people,
but I'm not talking about that. I'm not talking about that. I'm talking about the ratio of saturated to monounsaturated, which is the only thing that our physiology can really make a big impact on. We can easily turn a saturated fatty acid into a monounsaturated fatty acid, and we have stopped doing that because we need to stop doing that because we have so much PUFA. So the ratio of saturated to monounsaturated has changed in attempt to try and keep the melting point of our body fat relatively constant. So it's a protective mechanism, right? So we're trying to leave relatively more of the saturates in our body fat because they will keep it more solid. And if we were to desaturate that and have as much monounsaturated fat as we really ideally would want to, because that's the preferred fuel for mitochondria of the three types, then we would have too much melty fat. It would be, there'd be too much unsaturated fatty acids in our fat. So we're trying to keep it as saturated as possible to protect against all the PUFA. So you have to look at the ratio of the only things that we can control, which is the total monounsaturated and the total saturated.
Right, and that definitely happens. So polyunsaturated fat at certain levels absolutely does suppress the SCD1 enzyme, the thing that makes saturated fat to monounsaturated fat. The problem is at some further level of additional and longer polyunsaturated fat, they go up in parallel. The SCD1 starts increasing in parallel with increased polyunsaturated fat, and that's when you start to see, real obesity and real diabetes. That's what I've seen.
Well, it might vary in different tissues. Like if you're looking at the adipose versus the liver versus the muscle. So, I mean, you can't make a blanket statement about that. Were you talking specifically about adipose
I was talking specifically about adipose. Yes.
Okay. Yeah. So, I mean, we don't want to get too lost though. I mean, the big picture is that I think we should try to come to an agreement as to whether or not oxidative stress is a bigger issue than reductive stress. And I come down on the side of oxidative stress is clearly the problem. And I don't know where the reductive stress would be coming from these days, honestly. So, but I think if we can't agree on that, then we need to try to, because we have to pick one. We can't have both.
Well, you can't actually have both in the same cell at the same time. And that's one of those sort of unfortunate truths, right? Because what can happen is
what's driving it is what I'm saying.
Your pool. Right. And so I would argue that the specifically within the NAD plus NADH, it's ultimately the reductive stress that's driving it. But these are hard.
From? Because PUFAs cause oxidative stress, and we haven't changed our diet in a way that would cause more reductive stress.
Well, the PUFAs cause reductive stress in the NAD pool, but they cause oxidative stress in like the glutathione pool because, whenever you talk about, again, it's like the cell tissue type. Whenever you talk about reductive stress and oxidative stress, it depends both on the cellular subcompartment. Like, are we talking about in the mitochondria? Are we talking about cytoplasm? And then it depends on if we're talking about, NAD versus glutathione. So you can have both in the same cell. And that's, it's unfortunately it's hard, right? And it's confusing and it's hard to understand.
Well, it shouldn't be. I mean, we have to make it simple. Our job is to make it simple. And I think if we're paying attention to the wrong thing, we're making it more complicated than it needs to be. And so I think that, yes, there can be elements and like there can be basically little kinks in the system that is basically overall an oxidative stress problem that can cause here and there reductive stress, because it's a seesaw, right? So like you said, but the big picture is PUFA is causing oxidative stress. If we can't agree on that, then we can't agree on why PUFA is bad, why seed oils are bad.
Well, here's what I would say. I would say that if the biggest problem, if the problem, the overarching problem is oxidative stress, then the suggestion would be that if we were to take lots of antioxidants, we could make the problem better. But most experiments that I've seen don't suggest that's true.
That's not what the suggestion would be. That is a fallacy. So you have to understand that antioxidants, the term is so misused. It's not like resveratrol has this incredible antioxidant capacity.
Oh, I agree. I just mean like vitamin E.
And I'm sure you know this, but we have antioxidant enzymes, right? I think you've probably written extensively about one of them. We have many antioxidant enzymes that because antioxidation is you're taking something that's from a very high energy state and you're bringing it down stepwise to a safe energy level. And it takes more than one enzyme to do that. It takes more than one step. You can't just have vitamin C stepping in and solving all the problems, because it won't do that. It won't bring it all the way down to the proper energy level. And then vitamin C needs to be regenerated. And this is a fact that a lot of people don't realize, but every antioxidant compound like vitamin C can be pro-oxidant in a certain setting. So you can't make a blanket statement about like, oh, well, we can just take antioxidants like everyone does, like everyone selling supplements. I'm not saying you do, but everyone selling supplements says like, oh, you just need to take this massive best antioxidant supplement and then you won't have any issues with oxidation. And as you pointed out, those studies never bear out, and it's because it's based on a fallacy.
Could we just
in a healthy cell, the great bulk of the vitamin C inside the cell is in the form of dihydroascorbate. It's the oxidized form, which is active and protective. In reductive stress, you lose that function of vitamin C. You lose the oxidative function of vitamin C when the cell goes wrong and goes into reductive stress.
I'm sure there's a lot of consequences.
Can we just say that if you do anything which disables the electron transport chain, you are not going to be able to convert NADH in bulk to NAD+. So you will, however you damage the electron transport chain, you will end up with reductive stress. Whether you are damaging the electron transport chain with reactive oxygen species, or by acetylating it, or by adding ROS-derived polyunsaturated fatty acids to the proteins of the electron transport chain, there is a component which is ROS-derived, but the end result is reductive stress, because you cannot get rid of the NADH, or you cannot convert the NADH to NAD+. So that I would maintain that they both occur together. I don't think it's either or, and I think that reductive stress I would view as a marker of dysfunction of the electron transport chain. Just trying to see whether you can get these two things to be compatible. Okay.
Dr. Peat, do you agree with that?
Yeah, and it's reductive stress that turns on fat synthesis as well as lactic acid synthesis.
Right. Okay. So they're I mean, one could almost say they're two sides of a coin. They're going on at the same time.
What we're saying is,
and the process is
stress causes
what's going wrong is that you've got a dysfunction in the regulatory system that's balancing those two processes. Is that fair to say?
Yep.
And Dr. Peat, in your opinion, part of that, what's going on there is that you're not burning enough glucose.
Right.
Is that correct?
Right. I wrote a couple of newsletters on late 19th century experimenters with treating diabetes with huge amounts of sugar.
Right.
And what it's doing essentially is shifting the Randle cycle, lowering free fatty acids, stopping the interference with glucose oxidation, lowering fat production and lactic acid production, and getting respiration going.
Interesting. Now, one of the things that, if we're going to get this message out and we're going to convince scientists as to the validity of it, one of the things that we're going to have to explain better than I think we have is that proof of consumption is not a linear process, that you take a small amount and it has little effect on obesity, and then you take a large amount and it also has little effect of obesity. And if you take a large amount in a ketogenic diet, then it's actually seems to be beneficial from a solely obesogenic standpoint. Dr. Peat, do you have any thoughts on what's going on with that process? Because I've already seen there's a study that came out by John Speakman in 2018, where he looked at fat causing obesity compared to protein and glucose. And, he saw this clear pattern that, low levels of fat didn't produce much obesity, moderate levels of fat were the most obesogenic, and then high levels of fat, again, obesity started going down. Do you have any thoughts on what's driving that curve?
Yeah, when you have very large amounts of either fat or glucose, you're sparing protein. You're not oxidizing protein for energy, suppressing protein metabolism, and an excess of protein is a big problem that blocks thyroid function and oxidative metabolism, for example, and contributes to aging.
Tucker, and
yes,
you
sir.
were talking about ketogenic high puffer diets.
Right.
The famous Bioserves F3666 is 5% protein. And we don't know whether that controls body weight in the rodent model through ketogenesis and hypoinsulinemia, or whether it's purely driven by the extremely low levels of protein in the diet. So from what Dr. Peat was saying, protein levels matter, and the standard ketogenic diet model for rodent studies is massively hypoproteinemic.
Right.
It's borderline protein deficient.
Right.
So
because otherwise you can't get them to generate ketones.
That's the problem. Even worse if you supplement with normal levels of what do you package triglycerides up in to make VLDLs?
Glycerol.
To make the actual capsule bit.
The chylomicrons?
ApoB?
No, I'm just what on earth is the biochemical component?
Cholesterol?
Not carnitine, not carnosine. There's a
Cholesterol.
It's gone now. It's not going to come back. But they make it deficient in F366, so the liver cells can't export chylomicrons or VLDLs rather. So you end up with So you end up with an accumulation of triglycerides in the liver cells, which again is used to bump up the metabolism to ketones. So, but if you supplement with, hang on a second, it's on my desk.
Is it bile acid?
Carnitine? No.
No. Choline.
Right, of course, and that's why choline deficiency causes fatty
acids.
acid.
Choline deficient,
fatty
which
liver.
means you can't export fats from liver cells.
Right.
Hence it's keto. So it's a bit of a nightmare for trying to. I'll come back to what Dr. Peat said, was that you could be looking at the effect of not metabolizing protein. This is so. Our model's not very good.
Right. That would make sense, and it would explain why that diet often, but not always, causes fatty liver disease.
If you supplement with what did I say it's called? Choline, then it doesn't. You can basically take Bioserve and add choline to it, and no fatty liver. So
it's another non-physiological model for humans, effectively.
So. Yes.
Well, this discussion has been fascinating, and there's so much things to think about, but I do want to give our audience kind of a wrap-up question for each of you to tackle briefly. It's just kind of, I always like to make this, there's a lot of weeds we get into it, and I wanted that to happen. I want people to get into the nitty-gritty, and then for those who, they still want to know, okay, so what do you do about that? We've all explored different explanations as to why these seed oils and high PUFA products are linked to the rise of obesity. We've explained different theories about what's happening there. And you're not going to settle everything today, of course, but I do want us to think in a thought experiment at some point. If you were presenting a paper to Harvard, how would we organize all this information and come up with the best argument against the Harvard establishment for why they've gotten it wrong on these PUFA topics? But that's for another time.
Well,
yeah, go ahead. What's that?
I'll take that one. I would cite Mozafarian et al. 2011, which was produced by the Harvard School of Public Health, in which they show that the most fattening food by several fold is potatoes fried in seed oils. And then they turned around and tried to hide the fact. But their own data shows that seed oils are obesogenic. And if you compare in humans a potato fried in butter or a potato boiled to a potato fried in seed oils, they're not obesogenic, which kind of comes back to, Dr. Peat's fondness for glucose. Glucose alone isn't a problem. It's only a problem when it's accompanied with massive amounts of seed oils. So yeah, it'd be a very short presentation, I think. I'd say, here's your paper, guys.
There is at least one intervention study which can be interpreted in the same way as that. A single meal feeding study which looked at how much you ate before you stopped, which compared their only saturated fat one was potato, as opposed to the potatoes in PUFA were we call chips. What do you call them? French fries over there?
Yeah.
French fries,
okay.
Yeah.
Because chips, but basically, it could be viewed in those terms.
And I mean, there was another interesting paper that came out recently. I can't remember the author, but they showed that even one of Peter's favorite obesogenic diets, the Sirtuin diet, that if you blocked the effect of the oxidative products of the small amount of linoleic acid in that diet, right? The HNE, the toxin that dysregulates the Randle cycle and causes fat production. If you blocked that process, then you blocked obesity. And that's a diet with that's very obesogenic, but has a very small amount of linoleic acid in it. And at first you wouldn't think that the linoleic acid could be the driving factor of the obesity in that. And interestingly enough, the intervention that they used in those animals was a very common human polymorphism in the detoxification system for seed oils, the ALDH2 STAR2 mutation. So yeah, that would be my little chat to Harvard.
Well, I just want to wrap it up with everybody give a brief explanation as to what the best dietary intervention that you have found in your own research or life to deal with the obesogenic effects of seed oils and high PUFA consumption. Just try to give us a brief little explanation without going, we got to kind of wrap it up here, but I want to give everybody kind of a homework to explore. We've given the problem. Let's give a little bit of a solution for people who are wanting to deal with obesity in their own life, and you've explained seed oils causing it. So what do you do about it, in your opinion? Peter?
I got two points I would leave it with. One is that, oh God, it's going to go more than 2 points. Okay. The first is low carb eating sidesteps the problems of seed oils. Okay, that's the first. The second is that carbohydrate does not cause obesity in the absence of seed oils. The carbohydrate insulin model is incomplete. And long
does
term,
that mean if you're obese, having eaten seed oils, and you stop eating seed oils and you eat lots of carbs, it will not add to obesity? Or no, you're not saying?
Once you've cleared your fat cells of it, yes. You'd have to clear your fat cells of the linoleic acid first, but
that would be years, right, before you could eat high amounts.
That's why you should start with low carb,
right.
and then sidestep the problem to begin with while you sort out the problem that you've got. Mm-hmm. Okay, I think that those 2 points, I'll leave it at those two.
Dr. Cate?
I have this summarized on my Twitter profile as seed oils are death and deep nutrition is life. And deep nutrition is what every traditional diet around the country does to some extent still, but used to do. And we didn't have access to refined, it gets out the refined carbohydrates, which are empty calories. And then the rest of the edible world is really pretty much open, but you want to make sure you get enough protein. And the best way to do that, the most like evolutionarily, well, based on traditional diets, every traditional diet was pretty rich in animal sources of protein compared to plant sources of protein, and always in whole foods. Because we don't talk about it very much, but processed protein powders are also extremely unhealthy. And because the processed and the body they're partly, a lot of the nutrition in there is destroyed. It's not the way our body's supposed to absorb protein and so on.
Okay. Very good. Tucker?
Yeah, my Twitter hashtag for years has been LCL6, low carb, low six, for exactly the reasons that Peter described. I mean, you can clearly fix the problem just by lowering seed oils, but what seed oils induces is an inability to properly process carbohydrates. So clearly the most effective way to do it is to reduce both.
Brad Marshall?
Yeah, I agree with most of what's been said. I will say the other thing, my initial kind of hack for this, and I still agree with this, is to obviously, one, stop eating seed oils or other sources of omega-6, including things like commercial chicken and pork, which can be surprisingly high sources, and actually seek out foods that have a lot of stearic acid in them. And the reason for that is that well, there was a paper showing that supplementing stearic acid will, in fact, increase your rate of fat oxidation. And I think I have a guess as to why this is. When you look at humans or animals that are doing lipogenesis, new fat making, what you see is they tend to have a lot of PUFA. They tend to have a lot of monounsaturated fat, and what they have very, very little of is stearic acid. So I think the body sees fat with very low stearic acid as, oh, we should be making fat, and I think the body sees a situation with lots of stearic acid as we should be burning fat, because stearic acid is really the one thing that is like totally obliterated when those lipogenic genes are switched into the on position.
Dr. Ray Peat?
I think emphasis on body temperature should be greater. The PUFA lower your body temperature and turn on all of the inflammatory things that tend to keep your temperature down. And the paying attention to the digestive and circulatory events when you eat polyunsaturated fats or too much protein. The PUFA is already right in the bloodstream, turning down your metabolic rate by blocking thyroid transport and function, and by activating the stress-related estrogen functions. So even before you get to the cell, it's happening.
Very good. So what would you recommend dietary interventions for people with obesity caused by PUFA? Raise your body temperature?
Yeah, and stearic acid and sugar, for example, do that, and avoiding too much fish and poultry and pork. Those are the major foods other than the direct seed oils.
Very good. I want to let all of you guys have an opportunity to tell us where we can find your work, because I know a lot of people are going to want to do deep dives into your websites and so forth. So, Peter, where can people find your work?
Everything is on Hyperlipid, HYPER lipid. If you DuckDuckGo Hyperlipid and Peter, it'll come up as the first hit.
And Dr. Cate, where can people find your work, or anything you want to leave with?
Well, it's Dr. Cate, DRCATE.com. And there's plenty of information there about what a traditional diet is, deep nutrition.
Very good. Tucker?
My blog is yelling-stop.blogspot.com, and I'm very active on Twitter at Tucker Goodrich.
And Brad, where would?
Yeah, so my work is all on Fire in a Bottle. It's fireinabottle.net, but if you just DuckDuckGo or Google Fire in a Bottle, it'll come up. And I am on Twitter at fire_bottle. 90% sure that's my correct Twitter handle.
Dr. Peat, do you have a website or newsletter?
Newsletter is at raypeatsnewsletter@gmail.com, and the website is raypeat.com.
Well, very good. It's been a real honor to listen to all of you today. Wow, what a great time of discussion and learning. And I know our audience is going to be really thrilled to just see these types of mechanisms explored. This is something that you don't see happening in academia. I can assure you that. There's nobody doing this right now in universities having a kind of a symposium about seed oil mechanisms. So this is the future of academia in the 21st century. It's online now. So thank you all. Thank you all very much for joining me. And it's been a real treat. So thanks again.
Thank you, David.
Yeah.
Thank you very much.
Thanks, everybody. Thanks, David.
Take care.
Everybody.